Hi, today we will learn about Eliza, enzyme-linked immunosorbent assay. There are mainly four types: direct, indirect, competitive and sandwich Eliza. So first one is direct Eliza. Here usually in this to perform this Eliza we have to take something called as well. So usually in a microtiter plate you have so many wells. In direct ELISA, first you will add the antigen so that it will be coated by the antigen. Now you will add the antibody which is conjugated with an enzyme. So this antibody will bind to the coated antigen. And how to detect whether the antigen antibody interaction has occurred? For that we will add a substrate. So if antigen antibody interaction has occurred, the enzyme will act on the substrate and a color will be produced. So if presence of a color, that actually indicates the test is positive. In direct ELISA. Now in indirect ELISA, we want to detect the antibody presence in the patient serum. So for that initially we will add the antigen to the well. So the well is coated by the antigen. Now we will add the patient sera. So patient sera may or may not have the antibody. Suppose if they have the antibody, those antibodies will be binding to the antigen which is already coated on the well. Now which antigen we are going to detect? We should know the structure of that, which antibody we are going to detect. We should know the structure of that antibody, because here what we are adding next is, for that the antibody which we are going to detect, that antibody was injected into a gut. So what happens, considering this antibody as an antigen by the gut, they will produce some another antibodies, that is anti-human immunoglobulins will be produced. So these anti-human immunoglobulins which is tagged by an enzyme is now added. So already you have an antigen, then in the patient sera if the antibody was present, this antibody will be bind to the antigen coated on the well. Now you are adding anti-human immunoglobulins which is conjugated or tagged with an enzyme. So what happens? They will go and bind to the antibody. And if this binding has occurred, in order to detect that, we will add a substrate so that the enzyme act on the substrate and a color will be produced. So here also the presence of color means the test is positive. That means since we are detecting antibody, the antibody will be present in the patient sera. I think it is clear how the anti-human immunoglobulins is produced. The antibody which we are going to detect, we have to inject into the gut, such that in the sera of the gut we will get anti-human immunoglobulins which we are using for the third step. Okay. So that is about indirect ELISA. So in direct ELISA we used only one antibody. In indirect ELISA we have used two antibodies. Okay. Now in sandwich ELISA we want to detect antigen. So here initially we will add antibody. So that antibody will be coated on the well. Now you will add the patient sera. It may or may not contain antigen. Suppose if it contain antigen, this antigen will be bind to the coated antibody. Now the same antibody which we added initially, now we are adding again. But the difference is now the antibody is tagged or conjugated by the enzyme. Okay, so this antibody will also bind to the antigen. How is it possible? Because it is a polyvalent antigen. So it's able to produce bind with both the antibodies. Okay, so it's just like a sandwich. Antigen, and on the either sides you have the antibody. It is sandwiched in between two antibodies. Since the antibody which is tagged with the enzyme is bind to the antigen, now we will add the substrate. So what happens? enzyme act on the substrate and a color is produced. So the presence of color indicates test is positive in sandwich ELISA, and here we are actually detecting for the antigen. So antigen is present. Now in competitive ELISA, you can see here we are actually detecting the antibody. So initially we will have to add antigen so that antigen will be coated on the well. Now first of all we will add patient sera. Patient sera we are adding, and also we are adding the antibody which is tagged with the enzyme. So patient sera may or may not contain antibody. The same antibody which we are adding, but it is tagged with an enzyme. So in both we have the antibody. One is in the patient sera, the other one is antibody tagged with the enzyme. So actually there occurs a competition, that's competitive ELISA. So who will bind is actually if the patient sera contain antibody, that antibodies will be binding to the antigen. So what happens to the antibody tagged with enzyme? They cannot bind. They will remain free because all the antigen sites, they will be bind with the antibody in the patient sera. So what happens, even if you add the substrate, the enzyme cannot act on the substrate. The color will not be produced, because in here ELISA, after every step we are incubating and we are washing also. So since after this step, after this competition, when we are washing, what will be washed off is the antibody which is conjugated with the enzyme will be washed off, such that there is no more antibody present in that well which is tagged with the enzyme. So even if you are adding substrate, nobody is there to act on the substrate, so no color will be produced. But here the absence of color indicates that the test is positive. So what happens? What do you mean if there is no color? The antibody is there in the patient sera. Okay. Again you can see the direct ELISA. Here in the first case you can see we want to detect in the sample whether there is antigen or not. So initially they are coated. Now we are adding the enzyme conjugated antibody such that they binds color produced. In the second one, sandwich ELISA, you can see we want to detect antigen. So here initially you are adding antibody, then the sample which contains antigen, and again you are adding the same antibody which is tagged with an enzyme. So here also you get the color. And in competitive ELISA, which we have discussed at last, that was also to detect antibody. So initially you will add antigen and there will be a competition between the antibody present in the patient's sera or in the sample and also the antibody which is tagged by the enzyme. So in three cases color is produced, but in indirect ELISA color will not be produced if the test is